We're going to talk a little bit about the 3 types of surface receptors, and we have them up there. So we have G protein coupled receptors, catalytic receptors. I believe this is a receptor tyrosine kinase in the middle over here. And then we have ligand-gated ion channels, which we're going to talk about. So a little bit more about surface receptors. Cell surface receptors are usually transmembrane proteins, and they have 3 domains. So transmembrane, remember, just means that it's sticking on both sides of, it's sticking through the cellular membrane. And domains are just different parts. They're just different functional parts of each protein that have, different functions. So we have the extracellular domain that we spoke about before, which is also known as the recognition domain. And it's exposed on the outside of the cell, and this is what binds to the ligand. So this is what starts the signal cascade. It's sticking outside of the cell, hence the name extracellular. And it recognizes the specific ligand for that receptor. So that's also why it's known as the recognition domain. So the membrane spanning region is made up of hydrophobic proteins, and it spans the cell membrane is exactly what it does. So that's also a pretty self-explanatory name. And if you recall that the cell membrane is hydrophobic because it's a phospholipid bilayer, then that should be pretty intuitive to you that these hydrophobic proteins are the ones that are actually going to stay lodged in that membrane. So the makeups of each domain are a little bit different, and you could see that it sort of interacts, with the physiology and with the physical properties of the environment. So form does match function here. So the intracellular domain is the 3rd one, and it's also known as the coupling or the catalytic domain, and it's the active site, and it's located just inside the cell membrane. So we say active site as just a general, just sort of like a general term that some sort of catalytic activity is going on in there, but it varies greatly depending on what receptor you're actually activating, which we're going to talk about right now. So the first and most familiar one is probably the ligand-gated ion channels. So these are gated channels that swing open when the ligand is present, allowing for the passive flow of ions. So if you look to the right, this diagram, you have this closed, these are, we can look at these as the same channel, the same receptor. So if you look to the left, it's in a closed state. There are ions that are outside. Clearly, they're higher concentration on the outside of the cell than they are the inside. And it looks like the ligand for this specific ion channel is some sort of neurotransmitter. So that neurotransmitter is going to bind to the ligand-gated ion channel, and then it's going to switch open, right, like it is to the right over here. So when it switches open, it's a channel. So unlike the G protein coupled receptor that we were talking about before, there is an actual connection. There's a communication between the extracellular environment and the inside of the cell. So that's what a channel is. It just allows these ions to flow down their natural gradient. So it's not pumping, it's not using ATP to like pump ions in or out of the cell, but it is a channel, and it is allowing them to flow passively. But it can only allow these ions to flow if that neurotransmitter or whatever other signaling molecule attaches. So that's the most important part about ligand-gated ion channels, is that you can have a huge concentration gradient, but as long as this gate is not closed, those ions are not going to go through the cell membrane. So quick changes in ion concentration are very important in nervous and muscular functioning, as I'm sure you guys are all familiar with. And the most common ions that we see in these channels are usually sodium, calcium, and sometimes hydrogen ions and magnesium ions as well. And these receptors are different than the other 2 in that the transmembrane domain creates a physical channel that connects the cell and the extracellular environment, like we said before. And there are other types of ion channels, like voltage-dependent ion channels, but in terms of today's lesson, biosignaling, ligand-gated ion channels are definitely the most pertinent to this topic. So we touched a little bit upon G protein coupled receptors before, but again, GPCRs are the largest type of receptor. Upon their activation by an extracellular signal, the GPCR activates a trimeric GTP binding protein, which is also known as G protein. So we spoke about this a little bit before, but looking at this diagram to the left, this ligand is going to attach on the extracellular side. This is that binding domain that we were talking about before, the recognition domain. So when it does bind, this G protein, which is right over here, it was purple in the last picture, and it's actually green and purple in this picture, but it still is that trimeric alpha, beta, and gamma subunits. that whole protein is going to dissociate from the actual receptor whenever there's a ligand present. And then that GDP is going to be switched for a GTP, and then the G protein is activated, and it can go on and act as a second messenger, and it can, affect whatever change it is going to affect inside the cell. And it just continues that signal cascade. So, like we said, when the G protein exchanges a GDP, for GTP, it switches to its activated state. When the GPCR, that should say, is activated by its ligand, its G protein acts as a second messenger. It can open ion channels or act enzymatically on a target in the cell. So it has a couple of different roles, a couple of different ways that it can, affect change inside the cell. But this is a very important receptor. I think it makes up like half of all receptors that we target, like pharmacologically, in terms of treating disease. It's implicated a lot in pharmacology, and it's a very hotly studied field right now. Okay, so we also have a class of receptors called catalytic receptors. They're also known as enzymatic or enzyme-linked receptors. So these receptors have an active site on the intracellular domain where the catalytic activity may occur. So they're a little bit different than G protein coupled receptors, where if we go back and we look, this G protein coupled receptor, once it is activated, it allows this G protein to dissociate from the receptor, and they are now two different pieces. And that G protein is what travels throughout the cell and is the actual catalytic component of the GPCR. In terms of catalytic receptors, the catalytic activity actually occurs on the receptor, but it's in the intracellular domain. So they're often kinases. So whenever we learn about catalytic receptors, I know in like cancer biology classes, when you take them, you'll definitely learn about receptor tyrosine kinases, which I think are one of the more common catalytic receptors, and probably one of the ones that you'll see the most. So I believe this, yes, so this diagram that I have here to the right actually is a diagram of a receptor tyrosine kinase, which is just the more specific family of catalytic receptors. Yeah, they do what catalytic receptors do. So they phosphorylate other molecules. They actually phosphorylate themselves in a process called autophosphorylation first, and then that autophosphorylation is what activates the actual catalytic domain of the receptor tyrosine kinase. So usually it phosphorylates itself, and then the RTK will phosphorylate another molecule inside the cell, and then that molecule acts as the receptor tyrosine kinase's second messenger. So again, when we looked at the GPCR, the second messenger for the GPCR is the actual G protein. But if we have a catalytic receptor, it's going to phosphorylate some other molecule that's been in the cell, and that phosphorylated molecule is what is activated, and that is what's going to act as the second messenger, and it's going to continue the signal cascade. Which you could see when you look at the diagram over here. They're sort of, it's two separate receptor tyrosine kinases are actually sort of two separate proteins. And then when they are phosphorylated, they dimerize into one protein. And that's actually how they activate. So like the actual physical conformational change of the receptor tyrosine kinase is what activates its catalytic activity. And it allows for all this entire signal transduction.